Hi, everybody. My name is Christina, and I'm excited to introduce our speaker today. Susannah Burrows is an atmospheric modeler, so you may be wondering why is she at the land working group meeting, but we thought this would be a good match because one of the expertise of Susannah specifically is thinking about aerosol and other cloud nucleating aerosol in the atmosphere and how it links to different Earth system components. So that's what she's going to be talking about today. So hopefully it'll be a good introduction to this world of atmospheric aerosol and how it links to these other Earth system components. Susannah is an earth scientist at the Pacific Northwest National Laboratory, where she's currently serving as the aerosol deputy group lead for the EAMXX group, which is responsible for the development of the new global kilometer scale atmosphere model for the Department of Energy's E3SM model. So thinking about high resolution, Dr. Burrows earned her master's degree in atmospheric physics and her PhD in atmospheric science from the Johannes Gutenberg University of Mainz. And in 2018, she was awarded the DOE Early Career Research Program Award, and she worked on a project focusing on improving understanding of the sources and variability of atmospheric ice-nucleating particles, or INP, that we'll hear about today. And in 2013, Susannah Burrows also co-chaired an ASR workshop on new directions in atmospheric ice processes for the Department of Energy Atmospheric System Research Program. So she's also thinking a lot about clouds. Okay, so I'm going to pass it over to Susannah. We're very excited you're here. Anyway, thank you.
Great, thank you, Christina, and I also would like to thank the organizers of this working group meeting for the invitation to speak to the working group, and also to Christina for hosting my visit and suggesting in the first place that I maybe should come visit sometime. I am going to speak today mostly about the work from my early career project. I was encouraged that this would be of interest to this audience. But I think also just want to say a major interest of mine as well is kind of how can we bring the information that we're learning from observations and from field experiments into models to represent the processes. How can we identify which processes really matter for climate, and then find ways to represent those in models that can also connect between the land surface and ocean surface and the atmosphere. So in this project, our focus was to improve the fundamental level understanding of ice-nucleating particles. These are particles that have elements of their surface properties, so the chemistry and the physical properties of the surface, that allow them to facilitate freezing in clouds at much warmer temperatures than otherwise would occur. Without ice-nucleating particles, pure water does not freeze until temperatures below -36°C, but in the atmosphere we observe that freezing often begins at temperatures as warm as -20 degrees or -25, sometimes warmer. There is some variability in the temperature at which freezing starts, and we think that some of that variability is associated with variability in the concentrations and availability of ice-nucleating particles in different places. And so this is why we're studying this. The formation of ice in clouds is a particularly also a challenging problem. I'll talk a bit more about it, but it's very important for precipitation development. Most of the precipitation that reaches the Earth's surface develops in clouds that have an ice phase, and that's because once cloud droplets freeze, it accelerates the growth to sizes that are large enough to overcome the updraft velocities and fall to the surface. So that is a basic background. Before I get into too much detail, I want to acknowledge, of course, the funding agencies that have supported this work, the Department of Energy through the Early Career Program, ASR, the ARM program as well, and the EMSL user facility for chemical analyses. And the team that contributed to the work especially the 3 postdocs who worked on this, Gavin Cornwell, Isabel Steinke, and Aish Rahman, and many collaborators at PNNL and elsewhere, including Christina. So the kind of overarching, I've introduced already a little bit, the overarching question that's kind of behind a lot of my research in this area is, do we understand and then can we simulate the aerosol sources of INPs adequately to use them confidently in cloud schemes in regional and global models? So I'll talk a little bit about the background for that and then how we are trying to use ARM observations from the DOE ARM network and field campaigns to advance understanding, focusing a bit on sea spray aerosol, where there's past data from ARM and other field experiments, and then on agricultural dust, which we studied, or it was at least the goal to study as part of a field experiment under my project. And then I'll talk about kind of where do we go from here. So INPs impact ice formation in clouds in multiple ways that I want to explain very briefly. There are both mixed phase clouds and ice clouds can be impacted by INPs. In mixed phase clouds, the main mechanisms or processes that occur are immersion freezing, which is where a liquid droplet that contains an ice-nucleating particle freezes as a result of these surface interactions. But there's also a process of collisional freezing where just a particle that's not inside of a hydrometeor impacts onto or collides with a droplet, and this causes freezing, which happens at slightly warmer temperatures. This can also happen inside out if a droplet evaporates or the particle in it moves to the surface. This can also cause freezing at warmer temperatures. And then there's some other processes that we think are not as important in mixed phase clouds, but some of which are important, definitely important in cirrus clouds, including deposition nucleation, which is ice going directly, or water going directly from the gas to the ice phase. So these are important for precipitation as well as cloud radiative effects. I'm going to be focusing in this talk only on immersion freezing. So from here on out, I'm not going to say that, but everything I'm talking about is immersion freezing. The reason for focusing on that is because we expect it to be one of the most important, or possibly the most important, mode of freezing for mixed phase clouds. And we know from modeling work that model outcomes that we really care about, like the rate of Arctic warming in this example, can be very sensitive to the way that INPs are represented in the model. So this is from Ivy Tan's work showing that Arctic warming I think this was done in the CESM model, changed significantly between when she updated the INP scheme. So just these are 2 different INP schemes, and there's much more Arctic warming in this with this scheme on the right, and that was driven by feedback processes that contribute to Arctic amplification, such as the albedo and lapse rate feedbacks. So what this shows us is that at least the rate at which ice forms in those clouds is important, and in models that can be driven by the way INPs are represented currently. So where are these INPs coming from? If we want to represent their concentrations accurately, we need to know what their sources are, especially in a changing Earth system, like in the Arctic, where ice is melting and permafrost is melting. The sources are changing of INPs as well. So we need to know what the sources are. And so this is kind of an overview summarizing field experiment data for characterizing the concentrations of INPs as a function of the freezing temperature in air that is dominated by different sources in different places around the world. So you can see, for example, that there are biological sources that can contribute to freezing at very warm temperatures. And at colder temperatures, the dominant sources of freezing, or the predominant source is dust. But for any of these sources, there's 3 to 4 to 5 orders of magnitude of variability in the observed concentrations in the atmosphere, so a huge range of variations. And so those are large enough variations that it could matter to clouds. So how do we simulate those, and how do we get the sources right so that we can simulate the concentrations correctly? So we know mineral dust is an important source of INPs, but something that wasn't really understood until the last decade or so is that sea spray aerosol is also a really abundant source of INPs. And we also have been learning more and more about biological and biogenic sources as a source of warm temperature INPs, as I was mentioning before. So these three sources are ones that we know are important. And we know we need to be able to represent them if we want to simulate INP concentrations correctly. This figure is from work that I did actually just at the end of my PhD and beginning of my postdoc work, which showed that in remote regions where dust sources are really scarce, INPs predominantly could potentially come from sea spray aerosol. So this was our estimate, my best estimate at the time, based on kind of a study of literature values or literature studies of the sea spray aerosol and bacteria, biological particles that are contained in them, and their efficacy as INPs, and the global concentrations of sea spray and dust aerosol from models. I calculated the percentage contribution that we could expect to see in boundary layer air of sea spray aerosol versus dust, and to the INP population. And so that's what's shown here. And you see it just lights up, especially in the Arctic and in the Southern Ocean regions where you just don't have much dust, but you have lots of sea spray. And then we also know, another source that we also know is really important is soil dust. So I'll come back to the sea spray a little later, and I'm going to focus for the next part of the talk on the soil dust. So for soil dusts, we know from prior work, including some that was done at CSU, that's shown here, that dusts that contain organic matter from agricultural soils are much more effective INPs than mineral dusts that don't contain that kind of biological or soil organic matter material, whatever it is in there that's enhancing the freezing. And so in this particular study, they took a population of agricultural, or samples of agricultural soil, and separated out the INPs, and found that as the freezing temperature, as they looked at those populations and what percentage of them were freezing, the particles that contributed to the freezing had a larger fraction of those particles contained organics than in the total particle population. And then as they went to warmer and warmer temperatures, that effect increased significantly. So it seems like there's something happening at warmer temperatures related to this. And so this was one of a few studies that had been done in the lab demonstrating this connection between organics in the soils and freezing activity. And so the questions I wanted to be able to answer are, first, can we actually replicate this finding in the real atmosphere? If we observe dust that's in the air, in the atmosphere, will we find the same thing, or is this something that's kind of an artifact of the way that the sampling has been done and brought into the lab? We have seen this in other examples with INP research, that the ambient aerosol actually looks different in its behavior as ice-nucleating particles compared to samples people collect from the surface and bring into the lab. And so we wanted to see if this finding will replicate in the real atmosphere. And then also, another question is, do we actually need to know the organic and biological content of the dust to be able to predict its activity? And so in order to study this, I collaborated with colleagues at PNNL, particularly Ola Zeleniuk, and Gavin Cornwell was the postdoc who worked on this, to make use of a single particle mass spectrometer. So this is just here an illustration, not of the one we use, but it's just a nice animation to show how this works. Particles enter through an inlet, and then there are two laser beams. And the first laser beam ionizes the particle, and then and then the second laser beam measures it so you can tell how, you can count them and see how quickly they're moving. And then there's a measurement with mass spectrometry to produce spectra of M to Z ratios, which are unique to different types of particles depending on their elemental composition. And so we took PNNL's single particle mass spectrometer, the mini splat, to the field, to the DOE ARMS site at the Southern Great Plains in Oklahoma, as well as the PNNL ice-nucleation chamber, and combined these two measurements of the ice-nucleation and the particle composition using a pumped counterflow virtual impactor. So this is just a device that has, I'm minimizing this a little by saying just, it's actually a very critical piece of the experiment, but it's a device where you have a flow that goes against the flow of the sample, which deflects most of the particles that are smaller and have less inertia are deflected by this counterflow and flow out to the sides, and the particles that have greater inertia, the larger ones, can pass through. And so what happens is that we condense, in the ice-nucleation chamber, we condense water onto the particles, and then some of those droplets freeze. We bring them to a particular controlled temperature, say -25. Some percentage of them freeze. Those are the ones that have INPs that are active at that temperature. The ones that freeze then grow in size and through vapor deposition, and the ones that don't freeze tend to then begin to evaporate and become smaller. And so by the time they enter the PCVI, the ones that have frozen are significantly larger compared to the particles that didn't freeze, which are where most of the water has evaporated. And so we can separate them based on their inertia in the PCVI. And then the ones that had frozen and produced ice are passed through to the mini splat. We then evaporate the ice and then measure the single particle composition. It's a little bit complicated, very technically challenging experiment. We ran this for an entire month, and we sampled a total of 80 INPs, which is a record, as far as I know, for a single field campaign ever that has done this experiment. It's very challenging. And so that was one of the primary goals I had for this experiment, was to get a large enough sample size that we could have some statistically meaningful outcomes so that we can actually answer some of the questions that we had. So one question, a very basic one, is are dust particles more likely to be INPs? And we showed yes, they are in our results. So this is what we expect to see, but if we hadn't seen it, it would be discouraging. So in the total particle population, we see that actually less than 2% of the total particles were dust. The majority of the particles at this site are very small organic particles that come from condensed organic vapors. But among the INPs, approximately 25% were dust. So the dust particles were much more likely to freeze the droplets. And we also did a second experiment where we sampled the INPs onto substrates and did offline analysis using SEM-EDX, which gives us a kind of complementary measurement of the composition. And we saw the same effect there, that the dust is more likely to freeze. So here, the total particles, with this method, we saw about 20% of them being dust, and about half of the INPs. This method is not as good at seeing smaller particles, which is part of the reason why the numbers are different compared to the previous one. But what I really want to draw your attention to here is we also have this class of mixed carbonaceous dust particles. So these are particles where within a single particle there's both carbonaceous material and dust, which we can see from the elemental analysis with the EDX. And for those, that class of particles was only 1% of the ambient, but total ambient particles, but 20% of the INPs, which is like much more enhanced than even just the dust particles alone. So what we found is dust is a much stronger INP than the other types of particles in this air, but the the mixed carbonaceous dust particles are even stronger INPs than the pure dust particles. So this is the first time that I know of that this has been shown in the field. So we were able to demonstrate that result from the laboratory experiments does replicate in the ambient atmosphere in this experiment. However, it turned out that these particles didn't look like what we were expecting to see. What we thought we were going to see was mineral particles with some kind of like organic, sort of sludge, I guess, on the outside, some maybe soil organic matter attached to the outside. What the particles that we found actually look like under microscopy is the opposite. They have a core that is composed of carbonaceous material. And then they have these like spiky things attached to the outside that have lots of silicon and aluminum in them. Those are dust. So this is like completely backwards. It was a huge surprise to us, completely backwards from what we were expecting. And our best hypothesis as to what these are is that they may be wildfire particles that are generated so that where fires are emitting basically this, they produce these tar ball-like substances, and that either during the emission process the dust gets incorporated into them, or maybe more likely, once they're in the atmosphere, they collide with bits of dust and it sticks on the outside. So we saw these mixed carbonaceous dust particles being super active as INPs, but they weren't the type of particle we were expecting. So there's still more to learn here. And I also want to mention that Christina is actually one of the people who's done a lot of work on wildfire INPs. And I think you also found that when you measured ambient wildfire INPs, you saw evidence of dust playing an important role in the ice-nucleating activity from wildfires. So that's a really interesting area where I think there's a lot more that could be done to understand, how does that dust get into the wildfire smoke plumes, and then how much of that remains in the atmosphere and then contributes to ice formation in clouds. So that was so I'm going to switch gears now. So that was wrapping up the field campaign. But now I'm coming back to the beginning, talking about, okay, how do we bring these different sources together? So we've done a study to try to combine, produce kind of a more comprehensive understanding where we look at the dust INPs as well as the sea spray aerosol, and also biological INP sources, including fungal spores and bacteria. Pollen is another source of biological INPs that's been discussed a lot, which we didn't include here because the experimental data that we're using is from experiments where we can't really access the larger particle sizes that have pollen in them. So we didn't include pollen, but we have the fungal spores and bacteria. And so in this paper, we've tried to really bring these observations and models together. This is in a way that allows us to draw some conclusions about like how well do we actually understand the processes and simulate them now, and where are the biggest gaps. And so the approach we took is first to use observations from a previous field campaign to speciate INPs using two different methods. One, at the colder temperatures, we use the residual characterization method, which is effectively the same as the method I just described that we used in the field experiment at SGP. These observations, though, come from a coastal site in California. So at this site, we also have marine sources of INPs. So we use the residual characterization method at colder temperatures. At warmer temperatures, we can't do that because the number of particles that freeze is so much smaller at warmer temperatures. So if we can only get 80 particles at minus 30 degrees in a month, we're definitely not going to be able to get any at warmer temperatures where freezing is a few orders of magnitude lower. So we use a parameterization approach to infer and attribute the different types of particles at the warmer temperatures. So that's the observational analysis side. And then for simulations, we combined back trajectory simulations with maps of the surface sources that are both space and time dependent. So they have X, Y, Z coordinates. And we convolve those with back trajectories to basically project the particle source functions onto what we would expect to observe at the site where the field campaign was done as a function of time. So we can produce then a time series of simulated INPs based on the source functions at the site. And then in the last piece where we integrate it together, we compare those predicted time series with the observed time series in order to evaluate our skill in forecasting the INPs, not just the total INPs, but the INPs as a function of of particle type. And so, so the first thing we looked at is how well are we doing at the cold temperatures. At the cold temperatures, biological INPs don't play a very big role, but we did observe that, so this experiment did observe both sea spray and dust INPs. And this is the kind of model observation comparison. So the x-axis here is the simulated INPs, and the y-axis is the observed INPs. But we're getting really quite good agreement in both the variability and the magnitude for both of these sources. Sorry, I'm backing up. I misspoke. So this is the observed INPs from sea spray. This is the INPs calculated from the observed aerosol, not from the model, but from the observed aerosol surface area. So we get good agreement when we do that, which means the INP parameterizations, if we know how much of that particle type is there, the INP parameterizations give us the correct number of INPs from that particle type. Second step is that we use the predicted aerosol instead of the observed aerosol. And when we do that, our skill gets a lot worse, which is not surprising, but you can really clearly, by doing this, distinguish between what is an error in the model's ability to simulate the aerosol amount correctly at that exact time versus an error in the parameterization of how effective these particles are as INPs. So what we've basically concluded from this is our parameterizations of INP effectiveness are good. Getting the right amount of aerosol there is a struggle for the models. And so if we wanted to improve the simulation of these INPs, the thing we would want to focus on is improving the simulated coarse aerosol life cycle for sea spray and dust. And so that's at the colder temperatures. What about the warmer temperatures? So at the warmer temperatures, we found that the bioparticles were playing a really important role. And this is consistent with what's also been seen in other studies. For the bioparticles, we use a measurement from a fluorescence-based instrument. And the particles are classified into different groups depending on the wavelengths at which they fluoresce. So there's a fluorescent particle class that we call FP3. And this heat map here is showing which of several different variables, how well they correlated with the INP concentrations observed at different temperatures. And what you really clearly see is your eyes drawn towards this FP3 class, which correlated very, very strongly with the observed INP concentrations. So there's a very clear evidence of a link between INP concentrations in this warm temperature range down to about -18 and this bioparticle, fluorescent bioparticle class. So we know, so then from that we were able to parameterize the concentrations of the INPs from the bioparticles. For the dust and sea spray, we used our INP parameterizations and applied them to observed surface areas, but extrapolating to the warmer temperature range. And and then on that basis, we develop a partitioning of the INP concentrations within this temperature range between these three sources. So we have dust, so biological in green, and then the sea spray in blue, which is a very, very minor contributor in this temperature range. So the bottom line is that we find that based on this attribution, the biological INPs make up about 80% in this, of the total INPs in the warmer temperature range during this field campaign. And we think that is likely also true in a lot of other sites because many field campaigns have shown that there are correlations between warm temperature INPs and measurements of biological material. But to the best of our knowledge, this is the first time that this has really been broken down into an attribution to show like what fraction really is coming from biological. And then we took it to the final step and again compared the INPs that we get from the observations in these three classes versus what we can predict from the model simulations in these three classes. And so this figure is showing that as a ratio of predicted to observed INPs for the three classes. So if we're to the right of the the black line here is at 1. So if we're to the right of that line, the model is over-predicting that class of INPs. And if we're in the left, it's under-predicting that class of INPs. And so you can see that the sea spray aerosol is being over-predicted a bit and dust is under-predicted a bit. But the largest prediction errors are with the biological INPs. And so we think that that means, well, we think it could be a combination of having source functions that are not as accurate as we would like, possibly missing sources of biological INPs, or the parameterizations of the biological INP efficacy also have a lot of uncertainty, and there could also be shortcomings there. So there are a lot of places where that bias could come from, but I think the main kind of key finding here is that at warmer temperatures, the biological INPs are a major limiting factor on our ability to have a predictive understanding of INP concentrations in the atmosphere. And that matters because the warm temperatures are the ones that are going to freeze first in clouds. And so we believe that these warm temperature INPs are playing likely a very important role in triggering the initial stage of freezing, which then leads to a range of secondary ice production processes that cause clouds to glaciate. So the biological INPs may be determining kind of at what temperature that the initial freezing happens that results in cloud glaciation. And so we think those are super important. And this is just showing that we did also look at whether increasing the model resolution of the wind speeds would change our prediction or improve our prediction skill, and it had very little effect. So, so that's what we've done to try to bring all the pieces together of these different INP types and find out what we know and what we don't know and where are the biggest gaps in our knowledge. And so I just want to close for this talk with some kind of summary and forward-facing comments on this. One of the things that we know and that we showed in this work is that coarse mode aerosol, mainly the dust and sea spray aerosol, does explain INP concentrations that are observed in the atmosphere at colder freezing temperatures. So that's really important because it means that getting the sources of dust and sea spray right in our models matters if INPs matter. And there are still uncertainties in how we're simulating the aerosol life cycle. There's still room for improvement there. We know that biological INPs, or we showed that biological INPs dominated the uncertainties at the warmer freezing temperatures, which likely indicates that we need improvements in the fundamental process understanding of how those, which particles those are, how they're emitted, and and what happens also in the atmosphere after they're emitted that can change their efficiency as INPs. And so in addition to that, there are also some kind of fundamental open questions that need more advances in theory and observations and laboratory experiments to address. We discussed some of these in a review paper that Christina also contributed to in 2022 and at this ASR workshop that was mentioned. Those include the representation of the freezing process. There are different approaches, including deterministic formulations and stochastic formulations that have different sensitivities to updraft velocities and to time, differ in their representation of the time dependence of freezing. There's still some open questions around how detailed we need the representation of the freezing process to be. A second point is we believe that INPs are likely removed at a higher rate by precipitation compared to particles that don't act as INPs, because they may be more likely to be included in rain, but this has not been clearly demonstrated. So there are reasons to think it could be the case, but hasn't been clearly demonstrated, so that's a research question. And then a really, really important one is how do clouds respond to variations in INP concentrations? If you have more INPs, in the end, do the clouds really care? And that's a really tricky question to answer, partly because of what happens after the initial freezing occurs. So there are a range of at least 8 different mechanisms by which secondary ice can form in clouds after the initial primary ice formation. And those processes are likely to have a very significant impact on how sensitive the cloud phase and the cloud dynamics are to the concentration of INPs. So that's a major area where a lot more research is needed. For model inputs and constraints on INPs, I think we could consider developing an INP climatology that would be kind of a global climatology of the concentrations of INP sources or INPs in the atmosphere in different locations and different times of year. That would be really valuable for models that don't want to deal with prognostic aerosol representation in a lot of detail, which may be the case, for example, in global storm resolving models or kilometer scale models where computational performance is at a premium. And also, we need, I think, improved data sets for evaluating whether our simulations of INPs are adequate. So we did a very detailed study here, what I presented for a single field experiment, but also we would want to have more spatially representative sampling of, for example, dust concentrations in the atmosphere, and how they're evolving over time as agricultural practices change and as the climate is changing as well. So those are some of the things I think we still need. This is just an advertisement for our review paper, which discusses some of the issues, including changing surface sources of INPs in more detail and where all of the different uncertainties come from. And then once we kind of have that more fundamental understanding, we also have design choices to make in models about how complex our representation of primary freezing should be. So in the atmosphere, do we want to have ice nucleation be represented in a way that's independent of aerosols? So this is how most models have represented ice nucleation up until the early 2000s. This has some advantages. It's very simple and cheap, but also it prevents us from being able to capture spatial gradients in INPs that seem to be related to observed spatial gradients in cloud phase. So there are differences between the northern hemisphere, where we have a lot of dust, and clouds are seen to freeze at warmer temperatures, versus the southern hemisphere. Do we think that that's good enough, or do we think we need to have more diagnostic or prognostic INPs, so the linked prognostic aerosol? This is what a lot of models are currently doing, including E3SM versions 1 through 3, and including the CESM. This has the advantage that it allows us to look at dynamic responses. So if the dust sources change in the model, and because of land use change or climate, we can see how that changes INPs. And we can simulate the INPs based on dust if the dust is simulated accurately. But this approach is also susceptible to biases in the simulated aerosol life cycle. So if we don't have the sources right, or we're not simulating the removal rates through precipitation or sedimentation correctly, that's going to propagate into errors in the INP that are then linked to the cloud simulation. But this is what a lot of models are doing right now. And then a third option is that we could use diagnostic INPs that are based on more observationally constrained climatologies, as I was kind of mentioning before. So if we had really good climatologies of dust and sea spray, and then we use these parameterizations that we've shown are working in the real world for predicting INPs when we have the right aerosol amount, then we could have climatologies of INPs and link cloud formulations to that. This would be simple to implement. It's not so susceptible to biases in the processes that control the aerosol life cycle in a model. It's more realistic than just neglecting the distributions of INPs or aerosol globally. But it doesn't give us any way to look at dynamic responses between, say, land use change and INP sources very easily. So, but this could be a path forward for kilometer scale models like EMAXX, where we really need something that's simple and fast. So that's kind of where we're at, I think. I don't think I need to, I think I've got too many. I have had quite a few slides in here, so I think I've mentioned all these things already. I'll just leave this slide up here around kind of future directions. And I think there's a lot of interesting work still to do, including work on connecting to the surface sources better, particularly around the kind of biological sources and the dust that still can be, where there's still a lot we don't understand and that can be improved. So thank you for your attention, and I can take questions.
Okay, thank you, Susannah. Any questions from the group? And then, Paula, there are people online too. Okay.
Thank you. Very nice talk. I was curious about the biological sources, whether biogenic emissions, the way we do in the CLM with Megan, connect to the question of biological sources, or is that just chemistry? I'm not entirely clear, and so that's why I'm asking.
Yeah, thanks. It's a good question. So with Megan, we're talking more about the gas phase emissions of volatile organic compounds from plants, and those can contribute to secondary organic aerosol formation. It has been shown that secondary organic aerosol can weakly act as an ice-nucleating particle, and it's been suggested that in the upper troposphere, where it's very cold, that the secondary organic aerosol particles could be an important source of INPs because they could be very numerous, particularly in places like above the Amazon rainforest, where you have kind of a mega source of volatile organic compounds. Measurements in the upper troposphere so far have not shown support for that hypothesis, but I don't think that any measurements have been done in the upper troposphere over the Amazon, for example. So so it's possible it could be an important source there. Yeah. But in the places where they have been done so far, there hasn't been a lot of support for that.
So I have a question on your second to last slide. You had this sort of distinction of prognostic versus climatological ice-nucleating particles. And I'm just curious how important the high frequency variability is. I mean, think of like fire and dust as being highly episodic. Do you lose something if you move to the climatological that's important in terms of that high frequency?
Yeah, that's a really great question. So I think the answer is partly that we don't know. So one of the challenges that I talked about a little bit is that we don't, at a fundamental level, we don't entirely understand the processes that control the cloud response to INPs. So for example, so there are all these secondary ice production mechanisms. One example is that once you form ice, the ice crystals can collide with each other and shatter. And that forms more new ice particles, which then can grow through vapor deposition. And then you just can repeat the process over and over again and multiply the number of ice that you form. So because of these kinds of processes, I think we first need to know kind of like what is the background INP concentration, and then what is the variability on top of that, and then we need to know how do clouds respond to that variability. The answer to that is probably also different in different cloud regimes. And so I think the community is now moving towards more of a regime-based approach to looking at that question. So we may have a different answer to that question in the Arctic, where you have really long-lived, stable cloud layers and kind of a stable boundary layer conditions that can persist as supercool clouds for a really long time. But if you inject INPs into those from like a dust plume, for example, that could have a huge impact on these like super long-lived supercool clouds. Whereas if you're in the tropics and you have like a really, really robust tropical deep convection cell where there's probably so much air moving around between, including up to the upper troposphere, and with really high updraft velocities, you're cooling a lot. You probably are just constantly recirculating ice in those clouds. The INPs may not be as sensitive to INP concentration. So I think we need to kind of think about it in a more regime-based way in the future.
Yeah, so this all seems very complicated. And there's you mentioned a lot of things we could collectively try to do to try to move this forward. I'm trying to just get a prioritization thing in my head about, so I'm just going to give 3 examples of things that I think I might have interpreted that we could do and see what you say in terms of prioritization. So let's say on the dust, the carbonaceous dust and land use. So you implied that, you don't get, you need to get like land use impacts on, I guess, dust emissions to kind of get this right. But
Yeah.
Danny has shown most likely we're not getting that, Danny, in the back, that the models don't capture the impact of land use change and whatever soil degradation on. So that's a potential problem you could try to resolve. There's no carbonaceous dust, right? So we don't treat the fact that there's dust associated with carbonaceous. And then the third thing might be, okay, maybe it's not really the land processes, it's the atmosphere processes, and I don't know, is our atmosphere models accounting for these mixed dust, carbonaceous, aerosol properties? I'm guessing no, but you can tell me if I'm wrong about that. So if you had those 3 things to work on, which one would you tackle, or something else that's like the highest priority?
Yeah, well, so since this is the biogeochemistry working group, I think for the biogeochemistry community, yeah, I think getting the land use impacts on dust, figuring those out, I think would be a really high priority, and also that includes the agricultural dust sources. So knowing where the dust is in the first place from those agricultural sources would be really critical. And then as a second step, how much of this organic content do they have that is contributing to their efficacy as INPs. But then also, I think wildfires are a really important piece as well, because we are seeing increases in wildfires, and it's likely to continue in the future. So those could be, so even if the sources are in the mid-latitudes, so let's say over the southern Great Plains, we might not necessarily expect that the clouds in that area are extremely sensitive to INPs, because those are like mesoscale convective systems that are more dynamically driven than microphysically driven, but the particles that are emitted there can get transported to the Arctic, where the clouds might be more sensitive. So those agricultural dust sources, I think, are really important. And then after the emissions, there are also processes related to the chemical transformation of the aerosol that probably deserve more study, that can affect, like, how does the oxidation of the aerosol or condensation of volatile gases onto the surface affect the efficacy as INPs.
Can I just ask a follow-up? For the wildfire dust, is that like because of the winds being so strong? I think we have the ceiling mic. Is it, yeah, is it the winds being so strong that's kicking up dust particles from wildfires, or is it actually something coming from the outside?
I mean, it could be. I really don't know. I really don't know. I think it could be that. I also know that there's been some work showing that even after a wildfire has ended, that the dust emissions in that area are increased a lot for a long time after the fire. So that would be another source to consider. But the particles that are in the wildfire smoke, I would guess it's because of the high winds, but I'm not sure.
Yeah, just a quick follow-up on that. I think people from GFDL have done that carbonaceous dust by coupling fire with dust. So the mechanism is basically you have wildfires, and that burn out vegetation. It creates a shortage of soil moisture in a short term, and that exposed soil just creates more dust emission from there. So that's a mechanism that I think some of us here are thinking about adding in as well. Just a follow-up on the carbonaceous dust. Do you think it's important to add carbonaceous dust? Like actually, the question is, from your field campaign, I just wonder if they are externally mixed or internally mixed. If it's important to simulate something that's internally mixed of soot and dust in a model aerosol model. Is it something that we need to add?
So in terms of the mixing state of the aerosol, I think it probably does matter for the freezing. We're looking at that in some other work that I didn't show here, but I think the way in which it matters is going to depend a lot on so when you have a mixed particle, how is the surface of that particle different, or is it different from the particle that is not mixed? So if, for example, with these particles I showed that have the dust on the outside, if you had a particle that instead had dust on the inside and then it was coated with organics, that might have a very different ice-nucleation efficacy compared to the one that has the dust on the outside. So it might not just be as simple as knowing, you might need to also know something about the mixing morphology. Yeah.
Hello, I have two questions. The first one is related to like when you look at the fluorescent signal and you set to retrieve the concentration of different type of aerosol. I'm wondering like how did you conduct that part of field campaign? Do you put the fluorescent device such as waves to different layer of atmosphere to get that result?
So I will try to summarize. But if you're interested in the details, you can look at Gavin's paper where it's described how this was done. But essentially, the fluorescent particle measurement was done in parallel to the collection of INPs for sampling and in parallel to the characterization of the aerosol properties. So for that experiment, these are parallel measurements. And so we use the correlation between the fluorescent particle class and the INP concentrations to parameterize the bio-INPs.
Thank you. It's definitely important for me because we're also trying to do some field camping to sample the vertical layer of biological aerosols such as fungal spots. So maybe we'll also try to look at how to compare that concentration. And another question is from my personal interest because I'm most interested in the biological aerosol, and in your talk you have been mentioning like how important they can be, especially in the warmer cloud. In our previous study, like for regional model like WolfCam, we already tried to put pollen inside, and we are also continuously working on testing this model and put fungal spots inside too. So I'm wondering like, what do you think for you, like the current priority for including biological aerosol inside the INP simulation as well in the large-scale modeling framework?
Yeah, so I mean, I have a little bit of so my bias around what's worth putting into a model right now is that I work on the theirimpacts project now, where it's very operational in terms of our mission and goals for that project, that we really generally wait until something has been really proven to work in another context before we start thinking about putting it in, which I think is a little different. Like CESM is kind of a little more focused on exploring fundamental science hypotheses. We wait till someone else has done that, and then we add something new. So maybe that's a question that you can work on answering. Is I think that one of, well, I guess I will say, I think one of the really key uncertainties there is this process by which the spores and the pollen fragment and form small bits of pollen and spores, which then themselves can be INPs, right? So if you have their pollen concentrations are not very high, and they're really large, so they sediment really rapidly. But if you fragment one pollen spore to form 1,000 tiny INPs, that might matter. They might also stick to other particles and then make those particles more ice-nucleation active. It might matter. I think the challenge that I would, there's a challenge I think also for the observational community to come up with some methods to really measure that, to detect those fragments, so that once you have a model where you can, I think you have a model actually already where you predict them, how do you evaluate whether that model is giving you the right answer? And so there, I think there's an observation challenge to detect these very, very tiny protein fragments in the aerosol.
Ability, and when we put in the model, we found out like when we included these tiny particles, it improved the simulation of the convective system timing, and that can be like very interesting.
One very quick question, and then we'll end. It's quick. I think it's simple too. I'm just wondering, what about the biological aerosols like makes them freeze at warmer temperatures?
Oh, so that's a great question, and I didn't mention. Essentially, what we do know is that there are some biological particles that have macromolecules that are especially good at freezing ice. In particular, certain biological particles produce proteins that are very good at this, but also some other complex macromolecules like certain carbohydrates and lipids can facilitate ice formation. It seems to be related to their ability to form hydrogen bonds with water molecules at specific distances from each other that match the lattice crystal length of the ice crystal. So that seems to be the reason.
That's a great question enough on the scale of spatial and temporal scales of this problem. But thank you again, Susannah, for joining, and please continue the discussion over lunch. Thank you so much.
Thanks, everyone.